Gene mutations. Gene mutations are sudden changes in the composition of genes. And such sudden changes in the composition of genes is due to changes in the sequence of nitrogen bases. Changes in the sequence of nitrogen bases. There are three types of gene mutations. One is called substitution. Here there is replacement of a nitrogen base in the DNA by another one. So at any given place in the DNA molecule, a particular nitrogen base may be replaced by another nitrogen base. There are two possibilities here. One is called transition. Here a purine is replaced by another purine. For example, we know that adenine and guanine are purines in the nitrogen bases. So where adenine is present, it may get replaced by guanine, or a pyrimidine is replaced by another pyrimidine. In the DNA, cytosine and thymine are pyrimidines. So cytosine may get replaced by thymine, or thymine may get replaced by cytosine. So purine to purine or pyrimidine to pyrimidine. Another possibility is called transversion. Here a purine gets replaced by a pyrimidine. A purine may get replaced by a pyrimidine, or a pyrimidine may get replaced by a purine. Then it is called transversion. Then deletion. Deletion. In deletion, there may be loss of nitrogen bases, one or two nitrogen bases in a sequence. Loss of just one nitrogen base in a sequence in the DNA molecule can alter the code. That is called deletion. Insertion. Insertion. Insertion is the addition of nitrogen base. Addition of nitrogen base in a sequence. Addition of nitrogen base in a sequence. So there may be loss, there may be addition, or there may be replacement. These are the possible gene mutations. In gene mutations, we talk about frameshift mutations. Replacement of one nitrogen base by another, loss of one nitrogen base in the sequence, or addition of one nitrogen base in the sequence alters the sequence of nitrogen bases in the code. So the entire mechanism of reading the code, the entire frame of reading the code gets altered. So this is called frameshift mutation. So a replacement or deletion or insertion of a nitrogen base can alter the entire sequence of the code provided by DNA. When the code provided by DNA changes, the protein synthesis gets altered. We will study this in the next chapter in detail. So such mutations are called frameshift mutations. We also talk about point mutations. Point mutations, where there is a change of one nitrogen base. In the sequence of a gene, and sometimes, very rarely, there may be cross mutations. Cross mutations, where there is a change of several nitrogen bases in the sequence. But it is the point mutations that significantly bring about variations. Mutations can be, depending on their effect, mutations can be distinguished into same sense mutations, same sense mutations, where a change of a nitrogen base, same sense mutations where a change of a nitrogen base will not alter the codon. There may be a change of one nitrogen base in the code, but the amino acid ultimately coded by codon will remain the same. Another possibility is missense mutations. For example, UUU is a codon for the amino acid phenylalanine. UUC is also a codon for phenylalanine. Suppose in place of U, if C gets changed, it does not alter because both these code for the same amino acid. Therefore, the amino acid will not change. Then missense mutations. Here a change of one nitrogen base can change the amino acid. Can change the amino acid. For example, AUA is a codon for isoleucine. AUG is a codon for methionine. There may be a change in one amino acid. A gets replaced by G, but it codes for methionine in place of isoleucine. So amino acid gets changed. Therefore, the nature of the protein is changed. Hence, it is called missense mutations. The third possibility is nonsense mutations. Nonsense mutations. Here, a change of one nitrogen base can stop protein synthesis. For example, UGC codes for a particular amino acid, but if it gets converted into UGA, UGA is a nonsense codon. UGA is a nonsense codon. So in place of this amino acid, no more amino acid can be attached to the protein chain. So formation of the proteins in the chain stops. Therefore it is called nonsense mutations.